everybody. I am Dr. Aruna R, Assistant Professor in the Department of Life Science, School of Biological and Forensic Sciences at Christu Jayanti, deemed to be University, Bangalore, Karnataka. In this video, I will be discussing about the topic giant chromosomes. So what are these giant chromosomes? If we look onto some cells, the chromosomes are unusually large in size and they are also complex in structure. So these chromosomes, which are unusually large in size, are called giant chromosomes. These are so large that they can be seen under light microscopes. So what is the need of these giant chromosomes? Why are they so big in size? When there is a high demand for protein in the cell, then these chromosomes become unusually large in their structure to make more copies of RNA to meet the high demand for protein. Now, giant chromosomes are of two types. The first type is the Lampbrush chromosome and the second type is the Polytene chromosome. Now, if we look into the occurrence of Lampbrush chromosome, they are mostly occurred or found in the immature oocytes of amphibians and other vertebrates, while Polytene chromosomes are found in the salivary gland of larvae of Drosophila. Now, moving on to the next slide, Lampbrush chromosomes. These Lampbrush chromosomes are first observed in the salamander or amphibian oocytes by Fleming, and it was Ruckert who named them as Lampbrush chromosomes. So this Ruckert has actually observed them in shark oocytes. He named them as Lampbrush chromosomes because they look like the lamp brush that we use in ancient times. So this is a figure of the brush that is used for cleaning the lamp in ancient times. If we look at these both figures, it looks somewhat similar. So that is why Ruckert named them as these chromosomes as Lampbrush chromosomes. Moving on to the next slide. Now if we look onto the occurrence of Lampbrush chromosome, it occurs at the diploid stage, that is meiosis prophase 1 stage, in the primary oocytes of amphibians, molluscs and insects. Now why the Lampbrush structure is actually formed? Lampbrush chromosomes found in amphibians, oocytes can be seen during prolonged diploid stage, that is for many years. In these animals, development of new ones are outside the body. In this early stage of development occurs very rapidly so that the newborn can attain locomotion and can feed by itself. So this requires storing up of materials needed for early development even before the fertilization. So that is why in these species like amphibians, it gets accumulated and the genes for producing the mRNA are amplified and many types of RNA molecules are synthesized and stored for later use. So moving on to the next slide. Now, if we look onto the structure of Lampbrush chromosomes, as I said in the previous slide, they appear during the diploid stage of meiosis 1 and are characterized by their brush-like lateral loops, so which are the sites of intense transcription. Now if we look onto the components of the Lampbrush chromosome, the first component is the sister chromatid. Now we all know what is a sister chromatid means. So these are the two identical strands of DNA joined by the centromere forming the meiotic bivalent. Now it also has a maternal chromosome and a paternal chromosome. That is, two identical strands of DNA which are joined by the centromere forming the meiotic bivalent, in which each bivalent consists of one chromosome coming from the mother and other chromosome coming from the father. The next important component is the chromomere. Now this region is the chromomere. Highly condensed chromatin regions along with the chromosome axis act as an anchoring point for loops and they are called as chromomere. The next important component is the lateral chromatin loops. Now these are the extended loops of the chromatin that protrude from a chromomere, which is the site of the active RNA synthesis. Now the next component is the ribonucleoprotein matrix. Now what are these ribonucleoprotein matrix? So these are the dense coating of RNA and protein on the loops indicating active transcription. Then next we have the chromosome axis. So what is this chromosome axis? So this is nothing but it is a central scaffold formed by the chromomeres and connecting chromatin which supports the entire structure. Now if you look onto this structure, this is the entire Lampbrush chromosome. When only this particular part is zoomed, we can find that how it has been arranged like a Lampbrush. Now when only this particular loop is being zoomed, we can find that there is a chromatin loop, chromatid, chromomere formed from highly condensed chromatin and a chromatin joining adjacent chromomere. So this is how the Lampbrush chromosome appear like. Now moving on to the next slide. Now moving on to the functional significance of Lampbrush chromosome. If we look on to the functional significance of Lampbrush chromosome, the first important significance is that it serves as a transcription hotspots. So the lateral loops which we saw in previous slides, they are the actively transcribing RNA, especially mRNA, rRNA and other non-coding RNAs. And the second important functional significance is that it serves as a maternal RNA storage. So these RNAs are stored in the oocyte to support early embryonic development post-fertilization. And the last important functional significance of Lampbrush chromosome is gene amplification. So here if we look, there are certain genes which are amplified to boost RNA production, ensuring the embryo has sufficient resources before its own genome activates. So these are the important functional significance of Lampbrush chromosome. Now we are moving on to the next chromosome, which is a Polytene chromosome. Moving on to the next slide. The next special or the giant chromosome is the Polytene chromosome. So these Polytene chromosomes are also known as salivary gland chromosome because it is seen in the salivary gland of Drosophila. So these Polytene chromosomes are the giant chromosomes formed by repeated rounds of DNA replication without cell division, which a process called as endomitosis. So this is found in metabolically active tissues like the salivary glands of Drosophila larvae. They allow high levels of gene expression. So if we look into the history of these Polytene chromosomes or salivary gland chromosomes, it was discovered by E.G. Balbiani, and that is why it is also known as Balbiani rings or puffs. So what are these Balbiani rings or puffs? We will see in the further slides. These chromosomes were first discovered in salivary glands, hence it is known as salivary gland chromosomes as I said before. Now where do we see it? So it is found in the interface nuclei of some tissues of larvae of Drosophila. The metabolically active tissues such as the salivary glands, it will grow and it will increase in size. So in these cells, the DNA replication will occur, but the cells do not divide. So as the DNA replication occurs in the cell that are not dividing, the newly synthesized chromatids accumulate in each nucleus and are present as Polytene chromosomes. So what do we understand from this Polytene chromosomes is that the cell will undergo replication but it will not divide. Obviously when the cell undergoes DNA replication and does not divide, the chromosomes will appear in giant size. So that is what is happening in Polytene chromosomes. So the Polytene chromosomes are the giant chromosomes in which there are repeated rounds of DNA replication without cell division, and since it is found in the salivary gland of Drosophila, it is also known as salivary gland chromosomes. And the other name for Polytene chromosome is the Balbiani rings or puffs because it was Balbiani who discovered it. Now moving on to the next slide. Now if we look onto the structure of the Polytene chromosomes, the Polytene chromosomes are found in 6 arms from the chromocenter. Now this region is known as chromocenter. Why it is known as chromocenter means it is the centromere of all the chromosomes fuse in this particular region. Now this has a long arm which is an X chromosome and has a short arm which is the chromosome number 4. Apart from this, it has the right arm of chromosome number 3 and the left arm of chromosome number 3. It also has the left arm of chromosome number 2 and it has the right arm of chromosome number 2. Now if you look onto the right arm of chromosome number 2, we can see a region where the homologous chromosomes are separated. So altogether the Polytene chromosomes has a totally 6 radiating arms. Now if we look under the microscope, this is how the normal Polytene chromosomes look alike. A normal mitotic chromosome. Now moving on to the next slide. Now these are the important components of a Polytene chromosomes which I've already seen in the previous slide. Let's see a small recap. Now we have a chromosome center. This is the central region where the centromere of all the chromosomes aggregate. So it acts as an anchoring hub. Then it has an X chromosome, which is one of the sex chromosome. It appears as a distinct arm radiating from the chromocenter. It has 2R, that is right arm of the second chromosome pair, and it has 2L, which is the left arm of the second chromosome pair. It has 3R, which is the right arm of the third chromosome pair, and it has 3L, which is the left arm of the third chromosome pair. And at last, it has the smallest and the shortest arm, which is the chromosome number 4, often which is less transcriptionally active. Now, moving on to the next slide. Now let's look on to the functional significance of Polytene chromosome. The first functional significance is the banding pattern. Now if you look on to the previous slides, you would have observed that the Polytene chromosomes show distinct dark and light bands which correspond to the regions of condensed and decondensed chromatin. So these bands are used for gene mapping and cytogenetic studies. Now they have dark bands and interbands. Now 85 to 90% of the DNA band regions are bands and 10 to 15% of DNA has interband region. So 85 to 90% have banded region and 10 to 15 have interband region. Now these are the bands and the interbands that we see in the Polytene chromosome. Now when we look onto the Polytene chromosomes, I said it is also known as Balbiani rings or puffs, or it is also known as chromosomal puffs. Now what are these puffs or Balbiani rings? So these are the localized swellings in the chromosome arms where active transcription occurs. So they indicate regions of gene expression. The another functional significance of the Polytene chromosome is the gene amplification. The multiple chromatids allow massive transcription from many gene copies simultaneously, supporting rapid growth and development. So these are the important functional significance of the Polytene chromosomes. So in today's video, we had actually discussed about giant chromosome, like what is a giant chromosome and what are the two types of giant chromosome, and under that we saw what is a Lampbrush chromosome and Polytene chromosomes. I hope this video helped to understand what is a giant chromosome and also its types along with functional significance. So thanks for watching. See you in the next video. Thank you.